My name is Grace, and alongside my partners, Emily, Robert, and Lily, we are going to be talking about how DNA methylation works to actively change the stress phenotype in hyenas. After reading our article, we began to focus in on the specifics of DNA methylation that took place within the hyena population and connected this back to class. To understand what we were reading, we needed to understand what DNA methylation actually was, which is a mechanism that adds a methyl group to DNA to change the expression of the gene, which often occurs in response to environmental cues. This is shown in the hyenas with the correlation of increased maternal care and interaction when they are young to a positive level of global DNA methylation, which leads to a difference in the expression of affected genes, which in hyenas was shown to be the reaction to stressful events later on in life, which can be seen in cortisol levels taken from fecal samples. To guide all the research done in the paper following this information, the authors hypothesized that maternal care and social interaction in the early developmental stages leads to a difference in DNA methylation, which affects future stress phenotypes. This was then tested through the following methods.
To test the hypothesis, the researchers observed hyenas' maternal care in social connections and collected the blood and fecal samples from hyena population with known living conditions. They constructed 3 primary datasets for analysis, and then analyzed the data in 4 distinct parts in accordance with their goals. In the first 2 parts, they modeled the relationship between early social experience and the adult stress phenotype, indicated by FGCMs, which refers to the fecal glucocorticoid metabolites. And then they modeled the association between early social experience and global DNA methylations, and also the association between global DNA methylations and FGCMs. In part 3, the researchers used a formal mediation analysis to assess the extent of associations they modeled in the previous part, using global DNA methylation as mediator. In the fourth part, as a complement of part 3, they used genome-wide DNA methylation data as mediator.
By studying hyena cubs in the communal den period, the researchers concluded that neither maternal nor social influences are associated with FGCMs in adult hyenas. However, the researchers noted that for hyenas in the den-independent stage, higher network degree and strength are associated with lower adult FGCMs. The researchers found greater time spent in close proximity to the mother and more time spent nursing were both associated with higher global methylation. Grooming had no effect on global methylation. In the CD period, strength and betweenness were positively associated with global methylation, and in the DI period, network degree was positively associated with global methylation. In the subset of hyenas for which the researchers had data on maternal care or social network degree and global DNA methylation in adult FGCMs, the researchers found no association between maternal care, CD social connectedness, and DI social connectedness in FGCMs. Moreover, the researchers found no association between the explanatory variables and global DNA methylation. With respect to the lack of associations, the scientists did not carry out the last part of the mediation analysis. At a CPG site located at the human TSHZ2 gene, the researchers observed a positive correlation in grooming and a negative association in FGCMs with DNA methylation. At the TSKU, RAB43, 8AA, and ZSCAN2 genes, the researchers found negative association in grooming and positive association in FGCMs to DNA methylation.
Okay, so we know what the results are, but what do they mean? The first and third support the hypothesis that social connections promote and support healthy stress response among hyenas, as well as expanding their social network and increasing survival chances. The second and fourth explain that no evidence was found to support these ideas. Researchers urge caution, however, as other similar studies disagree with these findings, as well as noting that various discrepancies in lab could have caused error. Now, how does all of this relate to our class? Well, a large chunk of our learning has been on the unit of biological ecology. Ecology refers to the study of organisms and how they interact with each other, other close-by species, and their environment. Behavioral ecology focuses specifically on these species' reactions and how they optimize their survival and reproduction. In the study of hyenas, we can clearly view how this plays out. External interactions such as paternal care and community support influence DNA methylation. This influences gene expression, which then influences cortisol levels. Finally, this chemical change affects individual behavior to stimuli. We also discussed population ecology, in which we focused on life history trade-offs, referring to the negative relationship between survival and reproduction. A perfect example of this is as hyenas allocate additional energy to their offspring, their survival chances sharply decline. However, as the study outlines, more time and energy put into raising offspring strengthens their chances at survival through response to stressful stimuli. This is the works cited page for our project. Thank you for listening.